Bioinfocongress IV Dr. Sumru Bayın Age-Dependent Regenerative Mechanisms in the Brain. Dr. Sumru Bayın is a stem cell biologist with a long-standing interest in studying stem cell diversity and molecular regulation of stem cell behaviors in the brain. She completed her Bachelor of Science and Master of Science in Molecular Biology and Genetics at the Bilkent University and her PhD in Stem Cell Biology at NYU Langone School of Medicine in the laboratory of Dr. Dimitris Plekantomakis. She completed her postdoctoral training at Memorial Sloan Kettering Cancer Center in the laboratory of Dr. Alexandra Royer. Dr. Bayın has received a number of awards, including NIH Pathway to Independence Award, Sloan Kettering Institute Postdoctoral Research Award, and was selected as a 2020 Leading Edge Fellow.
Dear Dr. Sumru Bayın and participants, welcome all of you to our Bioinfocongress IV event. Dr. Sumru Bayın will give us a talk today on age-dependent regenerative mechanisms in the brain. At the end of the presentation, we will have a question-answer section prepared by our team and consisting of questions from our participants. Dear Dr. Sumru Bayın, welcome back.
Thank you so much, Nazlı. Thank you everyone for invitation, and I look forward to presenting.
Thank you so much. If you wish, you can start your presentation.
All right. Can you guys see the slides full screen?
Yes, hocam. We can see clearly.
Perfect. Hello, everyone again. Thank you for the fantastic organization and invitation. I'm really excited to be here. Also, thank you everyone who's tuning in on a Saturday, which is, in these stressful and upsetting times that we're going through. It is really nice to see all of you, and I hope you get a little break and enjoy the science you're going to hear these 2 days. And there's a fantastic lineup. So I'm really honored to be a part of the speakers for this Congress. So I am a group leader in the Gurdon Institute at University of Cambridge. So my lab is very young, just about a year old, actually. We just celebrated our first birthday. And in the lab, we're interested in the age-dependent regenerative mechanisms in the brain. And today I'm going to talk to you a little bit about, why are we so interested in stem cells, developmental biology, and how we can use these to address certain disorders or effectively develop therapies using this knowledge. So just to bring everyone a little bit on the same page, I would like to kind of give a brief introduction about stem cells. And as a stem cell biologist, I've been trained as a stem cell biologist, and I've been always fascinated by how can a single cell, like a fertilized egg, gives rise to us, very complex organisms, trillions of cells, more than 200 different cell types, and then how can molecularly a single cell know eventually what to make, right? And stem cells are crucial throughout the whole developmental biology process. And also, once we establish we are grown-up adults, maintenance of our bodies. So, we are not, we can survive throughout diseases and injuries and so on. So stem cells function throughout all of this process at different capacities while generating different cell types, but also using tissue-specific stem cells in the adulthood to maintain the organs and so on. And then how the stem cells do this is pretty critical. They are unique cells that have the ability to self-renew, so which means they constantly replenish themselves without losing the initial stem cell population, but also they're able to undergo a lot of proliferation through progenitor states and then commit to certain lineages, which eventually differentiate and mature to specialized cells like the neurons in the brain, that does the function of the tissue. And as I said, proper function of stem cells is critical for proper development, homeostasis, and something that we're really interested in the lab is in the repair or after injuries. So without proper stem cell functions, we cannot repair any injuries that causes cell loss or damage to the tissue. So, and importantly, we're interested in the balance of these processes, because when the stem cell function fails, you either get degenerative diseases, or if it's overly active and they proliferate out of control, you get overgrowth or other diseases such as cancer. So understanding the balance of what regulates stem cell behaviors is very critical. So we're a neuroscience lab, so we're really interested and fascinated by the brain. And in the brain, there are multiple different cell types that works in orchestration to fulfill function. Obviously, I'm sure you're all aware of the neurons, which are the electrically active cells that transpass information from one and another to fulfill, higher order functions of the brain. But there are also other glial cells that are either oligodendrocytes that help them to conduct information faster, astrocytes that modulate their function, microglia, which is the immune cells of the brain, and adult neural stem cells, and all the niche components such as the vascular cells or other different type of cells that can be in the brain. And the question we're interested in the lab is that how is this different cell types generated? And again, the answer is through the stem cells that are either multipotent that can give rise to many of these lineages, or through different progenitors that are, we call lineage restricted. So at that point, they are still proliferative and have some stem cell capacity, but compared to their predecessor, it's a lot limited, and they cannot make various lineages. They can only make the lineage they're committed to. So I wish the brain was simple enough that there was just 3 cell types, but actually brain is one of the most diverse, one of the organs systems that has vast cellular diversity. Well, and our understanding, and I think today and these 2 days, you're going to listen a lot about this, our understanding of cellular diversity has changed dramatically over the last century. So initially, we would kind of look into cell shapes and understand how that may be important for morphology and function, and like classify cells based on those type of events, to now with the generation of over the past, I guess, decade now, single cell technologies and spatial transcriptomics and so on, we can now, even within the same cell type, get into molecularly diverse subpopulations within different regions of the brain and so on. And even within the stem cell compartments of the brain, we now know that there are multiple different molecularly defined progenitors. And today I'm going to talk to you a little bit about that. But from a stem cell biologist point of view and a developmental biologist point of view, in the lab, we are interested in understanding how is the cellular diversity established during development and how is it maintained. Therefore, we're really interested in a concept called lineage plasticity. So this is a little bit different than you may have heard synaptic plasticity in the brain, where neurons wire, reorganize their wiring and so on to modulate function. Here we're talking about lineage plasticity. And what I mean by lineage is, I already told you that, different stem cells, diverse stem cells function coordinatively together to generate different neuron or astrocyte types and so on in the brain during development. And this happens through committed progenitors. So if there's lineage plasticity, we argue that possibly different progenitors that are committed to different lineages may change their fate at some point and give rise to alternative progeny. So this is not really a very highly observed process in the brain. And what is more interesting for us is that, especially when there's an injury in one of these components, what are the compensatory mechanisms? Are there any compensatory mechanisms to begin with? And if so, what are the compensatory mechanisms and lineage, injury-induced lineage plasticity that is in the system to replenish the lost cells? And importantly, in the lab, we are really interested in understanding what are the molecular factors, signaling, or genes that are involved in these different processes so that we can modulate this to induce lineage plasticity upon injury. So why do we care about understanding this? So traditionally, central nervous system is simply put, cannot regenerate, right? So when we get injuries to the brain or neurodegenerative diseases and so on, there is no efficient new neuron production to replenish the lost cells. So there are many studies that are working on trying to overcome this. And one of these approaches are, for example, generating stem cells or neurons in vitro from pluripotent stem cells, or you may have heard induced pluripotent stem cells from patients and injecting them back in. And there's a lot of promising studies, for example, for Parkinson's disease on that front. But in certain regions of the brain, we argue that this may not be as efficient. For example, one of those regions is the cerebellum, which is on the back of our brains. It has actually majority of our neurons, so 80% of our neurons are in the cerebellum. And it has these very complicated, layered and foliated structure that just injecting a ball of cells or neurons, we argue, may not have as efficient effect upon injury. Therefore, in the lab, we're interested in understanding how can we activate the stem cells of the cerebellum. First of all, are there stem cells in the cerebellum or progenitor cells? And how can we activate them upon injury to generate new neurons? But there's a lot of knowledge from developmental biology throughout to the adulthood that is missing for us to do this approach. So today I'm going to explain to you a little bit about how we are aiming to tackle this question. So as you may have guessed now, we're very interested in a specific region in the brain, that is the cerebellum. And there are multiple different reasons of why we're interested in that. So first of all, cerebellum is important for our motor coordination, how we walk, how we, our posture, everything, how we move. But also now, through its connections to the forebrain, people have been every day finding out new functions for cerebellum in pretty much every other cognitive and social behavior. The reason we're particularly interested in the cerebellum is that it also develops a little bit differently than the rest of the brain. It develops later. So majority of the growth of the cerebellum happens postnatally, meaning after birth. So here you see an MRI, MRI of a mouse brain, where after birth up to 10 days, 11 days, and then you can see how the cerebellum is growing a lot, and it's starting to form foliations to hold on to the to increase the number of cells it has to maintain them in a small structure. So this is up to 2 weeks in mice and up to 6 months in humans. So it develops a lot later, and it develops outside of the womb, which makes it susceptible to injury around birth. And actually, cerebellar injuries that happen around birth are the second leading risk factor for autism spectrum disorders. So it is really important to understand how the cerebellum develops properly and whether there are regenerative mechanisms around this stage. So over the years, starting with my postdoc research and now in the lab, we're interested in understanding whether there is increased plasticity and regenerative potential in the cerebellum around birth. And indeed, over the years, my colleagues and I were able to show that this is true, and that the neonatal cerebellum is also unique in its capacity to regenerate very efficiently. And today, I'm going to show you some of those mechanisms and how we plan to tackle this knowledge and developing this to further possible therapeutic avenues. So a little bit introduction onto how cerebellum develops, and some key cell types that we will be talking about today. So as I mentioned, most of the growth of the cerebellum happens postnatally, meaning after birth. And all of these cells of the postnatal cerebellum, all the stem cells in the postnatal cerebellum are defined, committed to lineages early on in the embryo, and they are either generated from the rhombic limb, which gives rise to, in the end, all the excitatory neurons of the cerebellum, which is a subtype of classical main subtype of neurons that fire in a positive direction. And the ventricular zone gives rise to all the inhibitory neurons, and importantly, all the glia, all the support cells that I had mentioned, such as the astrocytes, and a special cell type called Bergman glia, which is an astrocyte-like, but it has a special shape that is unique to cerebellum, which we're going to talk about it in a bit. So all these adult cell types, they need to undergo a lot of proliferation during the postnatal period, and then this proliferation is maintained by, is done by different progenitors of the postnatal newborn cerebellum. So one of this is the granule cell progenitors, and they proliferate on an outside layer of the cerebellum here, and then they migrate inside to give rise to granule cells, which are excitatory neurons. So one of the questions we were interested in the lab was that the nestin expressing progenitors, which give rise to the postnatal inhibitory neurons and glia, what are the lineages that are generated from the cells? And we knew they were diverse subpopulations of nestin expressing cells. So what are those? So I'm going to summarize because this is published, but that we were able to show that there are multiple nestin expressing cell subpopulations, and then they were defined by whether they were gliogenic, meaning eventually give rise to Bergman glia and astrocytes, or neurogenic, that give rise to inhibitory neurons. So they are lineage committed in the postnatal stages, okay? And then we were able to show that these guys were marked by either HOPX or ASCL1, which is a transcription factor critical for neuron production in the brain. So one thing to note, which is going to be very important, and that's why I put this dotted line here, is that if a cell type is from the rhombic limb, and in the postnatal case is granule progenitors, it cannot give rise to any of these ventricular zone-derived cells. So the progenies here that are defined as ventricular zone-derived or rhombic limb-derived is very lineage committed in the development. There is no lineage plasticity under normal circumstances between these cell types. But what we have done, and this was started with a previous postdoc in the lab, is that when we perform injury to the proliferating granule cell progenitors, and I'll explain you the injury in a second, we surprisingly observed that now the gliogenic HOPX positive progenitors, and actually a specific subpopulation of these progenitors that reside in the special layer called Bergman glia layer, due to the cell types they give rise to eventually, is able to now change its fate and give rise to the cell type that is affected by the injury, which is the granule cell progenitor, and then eventually give rise to the excitatory neurons. So this is kind of a classical definition of what I explained to you by lineage plasticity, that these cells that normally never make these 801 positive neurons is now with injury making this. And this process is called adaptive reprogramming. So what is adaptive reprogramming? It is a complex multi-step process. So when we kill the cells, and here we use irradiation because it selectively kills these highly proliferating granule cell progenitors, then what happens is in a day, granule cell progenitors die, and then this somehow stimulates these nestin expressing cells that are in the Bergman glia layer because they have these processes. And then these cells first proliferate, then start migrating to the site of injury. And then as they reach there, they start turning on granule cell genes, all right? So we understood a lot about molecular, on cellular level, how are these cells responding to injury, meaning, increased proliferation or migration and so on. But when we started in the project, we did not know much about the molecular events that take on here. So to do this, we took on a single cell sequencing approach to understand basically at all these different stages of adaptive reprogramming, what is driving these processes, which genes or pathways are involved in this. So before I get into those results, I want to give a brief introduction to those who may not be familiar with single cell sequencing. So it is a very powerful technique that allows us to identify basically on cellular basis, on single cell basis, obviously, hence the name, the transcriptome, or based on other approaches, for example, the chromatin accessibility of individual cells. And to do those, you basically join a single cell with these beads that have particular barcodes in them that you're able to selectively identify the single cell based on which barcode it is attached to and all the individual mRNAs coming from that cell in these little lipid droplets. You lyse the cell once they're in the lipid droplets with the beads, and then you perform reverse transcription to get cDNAs from the cells, and then you collect the barcoded cDNA. So each cDNA will have a unique barcode to designate as a novel molecule, plus the barcode that tells which cell it is coming from. And then you carry on with sequencing. And in the end, and I'm sure you guys are now familiar with more of these graphs, you kind of get graphs like this, which is either done with certain levels of dimension reduction, that each dot is one cell. And then after certain computational algorithms that I won't get into in detail, first of all, because I'm not an expert at them, but based on their similarity in their gene expression, grouping cell types together so that you can identify, okay, in my sample, there's 8 cell types or 8 cell states that's doing this and that and so on. So we took on this approach in collaboration with Peter Sims and Doğukan Mızrak at University of Columbia University to identify how is adaptive reprogramming occurring. So we sequenced 19,000 cells from postnatal day 5 nest in CFP cerebellum. So this means that we performed injury at day 1, which is what we always do injury, and then 4 days after we collected cells. And the reason we collected it 4 days after is that this is a stage where we see all these proliferation, migration, and transdifferentiation events occurring together. And we did this in a transgenic mice, which allows us to identify the nestin-expressing cells through a fluorescent reporter. So we got lots of cells from the whole cerebellum. We pulled out our nestin-expressing cells based on the CFP expression, the fluorescent marker expression, and then we did iterative clustering with or without injury to understand what are the changes in that. So we were able to observe our gliogenic NEPs and neurogenic NEPs and how they change with injury. So I'm going to summarize these results because this is all published and I want to talk a little bit about more unpublished stuff. But what this allowed us to understand that when we started investigating the injury-induced changes in our NEP populations, we started observing that the neurogenic NEPs, the distinction between the neurogenic and the gliogenic NEPs, kind of started graying out a little bit. And then we observed increased ASCL1 expression in the gliogenic NEPs. And this was surprising because ASCL1, which is, I told you, a transcription factor important for inhibitory neuron production in the cerebellum, is also never involved in the ATO1 excitatory neuron production. So it didn't really make sense why these guys would turn on ASCL1. So we wanted to investigate this further, and then we started looking into genes that are likely to be expressed with ASCL1 with injury, which is you see on this side of the graph. This is a probabilistic analysis of 2 genes being expressed together in the data. And you see likelihood of genes to be expressed with ASCL1 in the injured cells. And then we observed that in the control case, all the genes we get were restricted to the white matter, which where the interneuron progenitors are. But upon injury, we started seeing genes that are known to be expressed in the Bergman glia cells. And this is the cells that induce lineage plasticity to replenish the lost cells. So we hypothesized that actually the Bergman glial NEPs during adaptive reprogramming to ATO1 positive granule cells, they undergo through an ASCL1 positive transitory state. So we wanted to confirm this since this is this hypothesis set us as an interpretation of the single cell data. So we started performing, first of all, RNA in situ, so where we look at ASCL1 expression. And indeed, ASCL1 itself is also increased in the injury in Bergman glia layer. So we wanted to do as a developmental biology, just gold standard experiment to see if these cells do indeed give rise to granule cells with injury. We performed fate mapping. So when we perform fate mapping, just to briefly explain to the folks who may not be familiar with it, we use an ASCL promoter-driven Cre recombinase that translocates into the nucleus when we inject a drug called tamoxifen and removes the stop codon in front of an fluorescent protein. Therefore, when the tamoxifen is injected, cells that express ASCL1 will turn on red. And any cell from that point on that is generated from that red cell will maintain the red because we're modifying the genome at that point. So in the control case, when we do fate mapping and look a few days after, basically just looking at what is initially labeled, all the labeling we get is in the white matter, which is where the interneuron progenitors are. And then occasionally cells we get in the Bergman glia layer, which is the injury responsive layer, are the cells that are migrating out to form the interneurons. But when we look at injury now, as we hypothesized, we are now getting these new cell types being labeled, which has these projections to the outside of the cerebellum, like the Bergman glial express NEPs that generate Bergman glia, but now they actually are expressing ASCL1. And when we look at later, so we do injury, we label the NEPs, and then we look a couple weeks after to see in the end whether they were really able to establish granule cells. And then here you can see the quantification, which is easier to interpret. Only with injury now, we're seeing granule cell progenitors produced from ASCL1 positive cells, okay? We also see an increase in interneuron production because of the delay in development when the injury is happening. And then this is important. We did not see any increase in the Bergman glia labeling with injury. This means that once the cells turn on this transitory ASCL1 population, they are really committed and they cannot go back to make Bergman glia, okay? And therefore, we thought ASCL1 is required to inhibit this fate so that the cells can take on this new fate with injury. And then we also observed increased astrocyte production, which we don't know exactly what to make out of it yet, and curious to understand further. But this is kind of an interesting process because ASCL1, as I told you earlier, has been used heavily to induce neurons. And people actually are overexpressing ASCL1 and other astrocytes in the rest of the brain to generate neurons after injury. But this experiment was pretty exciting for us because in this case, cerebellar cells, cerebellar progenitors, are able to turn on this gene on its own. We're not doing anything. The animals regenerate very well, and there is some inherent capacity of these cells to carry on adaptive reprogramming and lineage plasticity. So in the lab, we're now trying to understand what is this inherent capacity. And importantly, as you can see, and I pointed out many times today, ASCL1 is normally involved in a completely different process and cell type production, where with injury now it's different. So in the lab now, we're trying to understand how is the ASCL1 functioning to establish this new lineage with injury. And to do so, we're looking into also not just the RNA of these data, but we're performing the single-cell attack sequencing, which takes into account the open chromatin regions, where you can infer what is epigenetically open as opposed to epigenetically silenced, which is a way of stem cells deciding which lineages to make or not, so that we can understand how the ASCL1 function in this injury-induced state versus the developmental state is different. So the data you get from this is pretty similar, but now, again, each dot you see is one cell, but the clustering is made based on inferred expression, based on the open chromatin, whether it's close to a gene or not, a promoter region of a gene or not. And then we were able to show that, and we're investigating this further. For example, with injury, there are very specific regions that just opened up that has binding motifs for ASCL1, which may be important for the lineage plasticity. So we're right now trying to understand this, but hopefully just to take a little breather and summarize a little bit here that we are pretty confident now that the neonatal cerebellum is pretty unique in its regenerative potential, and then it is a very good system to study lineage plasticity, which is not very commonly occurring in the brain and elsewhere. So one other question that lab is really interested in based on these results and the uniqueness of cerebellum is also, you know, neonatal cerebellum is unique and regenerative. Does this mean can the adult cerebellum also regenerate? Maybe it was something we're missing. And when we say brain is not regenerative, we haven't really truly explored the adult cerebellum. So just for those who may be more familiar with the neuroscience and neural stem cells, cerebellum is not a classically neurogenic zone. So it doesn't have ongoing neurogenesis in the adult like the subventricular zone or hippocampus. So we were first wanted to investigate whether there are stem-like cells or any stem cells in the cerebellum that may be responsive to injury. So we started looking into our favorite marker now that marks the NEPs, which is nestin. And we were able to show that there are actually rare nestin-expressing cells in the cerebellum. And interestingly, most of these cells, about 75% of them, are in the Bergman glia layer. And remember, this is the layer that has the cells that is able to regenerate in the neonates. And then these cells do not express mature Bergman glia markers, which is S100 beta. So they do seem to be more undifferentiated. And they are able to fulfill certain stem cell criteria, one of which is single cells' ability to grow into these round fat spheres, which is a readout of they're able to self-renew. So they are indeed able to perform this assay better than the CFB negative cells of the cerebellum. And then finally, when we do fate mapping, which is our way of understanding what do these cells give rise to, they fulfill the lineages of their neonatal counterparts, so they are gliogenic, so they don't generate neurons, but just give rise to Bergman glia and astrocytes when we do fate mapping. So this was really exciting at the time because, you know, there is an undifferentiated looking cell type in the brain. Can this regenerate? So we performed experiments to the adult brain. So we did injury, and then we were excited to see that actually with injury, these cells are able to know that it is injured and are able to increase the production of astrocytes and neurons. But they are not proliferative and no new neurons is generated. So we think it's coming from a different cell type, which I'm not going to get into it today. But one thing right now we're in the lab we're interested in is, okay, there are stem cells in the adult, but why can't it regenerate, right? So the neonatal SNPs I showed you are able to start from the stem cell state and go all the way to the neuron production with injury through these different variation of steps. Whereas adult NEPs, they know there's injury, they do something, but they are really blocked starting from proliferation step. So in the lab, we're interested in understanding how can we push these adult NEPs to regenerate neurons upon injury. So our hypothesis and our approaches is twofold. So in order to be able to facilitate regeneration, we believe that we need to understand the cell intrinsic and extrinsic mechanisms that first allow regeneration in the neonates. And I already showed you some of the approaches we're using for this. And importantly, inhibit regeneration in the adults. So how can we make the adult non-regenerative nerves become regenerative is in the end our goal. So to do this, we hypothesize multiple different mechanisms that act together. One is once the development ends, the developmental signaling is reduced, so these cells are not actually as stimulated to proliferate or differentiate. Secondly, once the stem cells commit, they usually silence other pathways so that we don't get the differentiation or tumors or aberrant reprogramming, so that the adult tissues maintain properly. And then finally, the environment may be permissive to regeneration, pro-regenerative. And for example, the immune component in the neonatal and adult can be different. And we're investigating that in the lab, but I'm not going to talk about it today. So first, we wanted to investigate whether if we increase developmental signaling in the adult NEPs, can we induce more regeneration? So to do that, we knew sonic hedgehog pathway was required for regeneration from our previous studies in the neonates. So in the adults, after injury, we activated sonic hedgehog and look into the NEPs to see if we observe any increase. And indeed, injury plus sonic hedgehog activation increased the number of adult NEPs significantly. But this did not lead to any proliferation and neuron production. So what happened basically was we were able to push this tiny bit further, but again, there was some internal blocks. So now in the lab, we're thinking that we need a combinatorial and sequential approach where, for example, we first induce proliferation, then plasticity, and so on and so forth to really fulfill the whole lineage steps. To understand, though, we still need to understand how of all these different steps are regulated molecularly. So we're going back to single cell sequencing that I mentioned to you. And we now are, the previous data I showed you was only just one time point, but we're now looking into all the different stages of adaptive reprogramming to understand how the lineages are made and how it changes with injury. So you can see a sample data here from all these time points that varies a lot with age. Like, for example, we get the granule subprogenitors at later time points, which is a later injury-induced cell type, and so on. And then we are able to identify all our favorite cells from this data to investigate further. So as I said, we're really interested in understanding the lineage dynamics and the injury-induced signatures. So to do this, I want to give you just the final idea of type of analysis we're interested in. We are pulling out our HOPEX expressing NEPs and ASCL1 expressing NEPs, reclustering them, and then looking into how does the HOPEX expressing NEPs change with age and injury, vice versa. You can see like we're looking into the population dynamics over time. And then importantly, we perform an analysis, one of which was called RNA velocity, which orders all these single cells based on pseudo time. And for example, when we look at it for the gliogenic lineage, we can then infer different paths that the stem cells go through to make astrocytes or Bergman glia. In this case, you can see two paths, one for Bergman glia, one for astrocytes, to really understand what is driving, at this point, different lineages from these cells. And then the other type we're looking into is also same with the neuron production. Again, how the different paths that these single cells take to generate in the end neurons. And you can see, for example, we start with the progenitor here, and then as it matures, it becomes the immature interneurons that is labeled by PAX2, and then the algorithm is able to detect the directionality of this. So now we're interested in these differential nodes. Where are these? What are the molecular mechanisms that making the cells to become more mature, and so on and so forth. And then finally, as I mentioned, we are getting a lot of idea from this data, and we are already able to, I'm not going to get into detail, but get some ideas about possible mechanisms involved in all of these steps, and particularly the lineage plasticity, which we know is really inhibited in the adult. So we're trying to understand how in the adult we can induce this, and what is the signal that inducing this transitory state that is required for regeneration. So to do those, we're also using in vitro models from mouse NEPs in vitro, and then differentiate them into neurons and astrocytes to study how we can push them to one lineage or differently. And then finally, last piece of data I want to show before we finish today is that the reverse side of the coin, the silencing of developmental pathways, and how we plan to study that. So to do that, we're looking into why the adult regeneration at what step is blocked. And to do that, we initially took on a chromatin-based approach where we're again looking at the attack sequence data of the NEPs from the neonates with or without injury, and then the adult with or without injury. So in the neonatal, here you see a volcano plot, a tornado plot that has the open chromatin region. So if a region is open, it's darker blue. And this is kind of the transcription start side of genes that are involved in neurogenesis and proliferation, all right? So in the neonates, everything is pretty much open because this is still an actively developing and neurogenic status. And injury doesn't seem to change much. But you can see in the adult, all the neurogenesis and proliferation pathways or genes seem to have closed chromatin. And thus we know is the stage that is blocked. And the injury is unable to turn on these genes. Whereas upon injury, for example, the glial differentiation and stretch genes, which is from our fate mapping, suggests it is able to partially turn on. So there is, besides lack of developmental signals that I had mentioned to you, there is active silencing of some of these stem cell programs in the adult NEPs, despite there is injury. So one of these programs is actually lack of MYC. MYC is a proto-oncogene that is really important for many processes, but also cellular proliferation and reprogramming, actually. So in the neonatal NEPs, we know that MYC is highly expressed, and you can see also the open chromatin around the MYC gene. But in the adult, it is inhibited, it is silenced, and with injury, it is unable to turn on. So one of the mechanisms we're testing now is that if we activate MYC, can we get more proliferation of the NEPs? So we use the transgenic mouse line that induces MYC overexpression. And you can see when we overexpress MYC, actually, selectively Bergman glial NEPs, and we identify them using SOX2 here, is able to proliferate where normally they're not proliferated, which was exciting. So we're hoping to now push this a little bit further to overcome the lack of proliferation. And to do so, we're collaborating with Catherine Wilson and Department of Pharmacology here to use actually modified RNAs, because, you know, we don't want to develop a therapy that can give you cancers down the line, because MYC overexpression is a very well-studied oncogene. So we're using modified RNAs, similar to all those RNA vaccines we all got over the last couple of years for COVID-19, that is stable, but transiently induces MYC expression in the cells, and then doesn't integrate into genome and cause any other problems. And we have some preliminary data that it looks very, very promising. So the goal would be to induce MYC overexpression transiently so that we can overcome this proliferation hurdle we're having, and then build upon it on additional signals, for example, whatever that induces ASCL1 expression. So with that, I'll summarize and then finish so we can talk to questions. So hopefully today you guys were excited about, you know, importance of stem cells and their function during development, but also how we can utilize them for regenerative therapies, and that neonatal cerebellum is a very powerful system to investigate some of these molecular biology behind stem cell behaviors, and how the regulation of their self-renewal, importantly lineage plasticity and differentiation, is can be harnessed for future studies. And then in the lab, in the next, some of our primary goals is to translate our findings to human biology, but also understand whether pro-regenerative signaling is involved in tumorigenesis, and whether other regions of the brain have similar age-dependent regenerative processes. So just a little bit of acknowledgements. This is my lab. Some of the data I showed you today was generated by Jens, the single-cell data, and Jada has done some of the mixed studies, a list of our collaborators. My postdoctoral lab is where most of these studies have been started, and actually some of the single-cell data has been generated, and our funding agency. So we're always looking into excited and talented people to join our lab. So if you were interested in any of this, please feel free to get in touch with me, and I'll be happy to take any questions in English or in Turkish, whichever you're comfortable with. Thank you.
Thank you very much, Dr. Sumru Bayın. I'm directing the first question. An increasing number of pro-aging and pro-youthful factors are being identified in the blood. To what extent do pro-aging and pro-youthful factors converge or act through different mechanisms?
Okay, yeah, that's an interesting question. Yeah, it is part of the, I guess, the cell extrinsic mechanisms we're talking about, right? So all the, there's a lot of old studies where, for example, you stitch together a young and an old mouse, and then the blood of the young mouse is able to rejuvenate the old mouse as well. So there's many different processes. So I think it is a little bit hard to say how do they act through different mechanisms, because they all in the end stimulate certain processes. But when we talk about aging, part of the mechanism is to prevent accumulation of, for example, reactive oxygen species, or like, you know, harmful substances in the cells and so on. Whereas some of the other mechanisms, which is what more we're interested in, is to stimulate new cell production. So it can be preventing cell from dying and functioning properly, or it can stimulate new cell production or tissue turnover. So those are two main mechanisms. But if we get into molecular mechanisms, I think there's so many different approaches that they can happen. And I need to clarify one thing. So we say it's age-dependent regenerative mechanisms, but it's also one thing I should point out. The point we see adult regeneration not happening is not really even an aged organism. It is adult, meaning right after development ends in the sense. So some of the regenerative mechanisms are really restricted to developing brain. So even if it's not, you're not aged in the sense like, you know, 20-year-old, your brain still doesn't regenerate, making new neurons. So it is more cell intrinsic in certain ways than an actual aged organism, if that makes sense.
Thank you so much. I'm continuing with the next question. Nowadays, do we determine whether NAPs in adult cerebellum can be stimulated to replenish cells lost due to the injury?
I mean, that's what we're working on right now. So we know these cells are able to sense there is injury. So what I mean by that is they increase in numbers, they become more stem cell-like, but again, they cannot make neurons, which is what in the end we need, because neurons are the cells that are dying. So that is our lab's goal. So to determine this, we're doing lots of fate mapping with injury for adult NAPs after injury to see if we can get more neuron production. So stay tuned. Yes, that's our goal. But we haven't made extensive number of neurons from NAPs yet. So that's why we're going back. That is exactly actually the reason. We're going back to the development to really understand how the the developing brain can regenerate so that we can replenish those signals in the adult, because right now they are unable to.
We appreciate for your answer. Thank you so much. I'm moving on to the next question. As an example on zebrafishes, young and elderly fishes are differentially impacted from stress, especially for young fishes. It can be turned to chronic stress, but regeneration ability isn't changed. Could we say physiological balance is highly affected by stress response and maybe after that growth and tissue regeneration?
Yeah, no, that's a great question. Again, so for example, there is a study, a lot of studies in the brain that exercise actually stimulates stem cells, for example, right? And then keeps them happy and neuron regenerating. So on the other hand, constant stress can let these cells also molecularly kind of gain epigenetic memory due to like environmental stress, chemical toxins, all sorts of things that will eventually prevent them from functioning as well. But again, coming back to our research, the adult NEPs in the cerebellum are, even default, are unable to make neurons. So we need to basically make them remember that when they were during development, know how to make this, and they should remember that and make it again. But yes, generally, I think regarding zebrafish brain, it is a great model, but in this case, it is not really the best model to compare our results to, because actually zebrafish cerebellum never stops regenerating, as the person has pointed out, and that actually there is constant proliferation in the cerebellum, specifically in the zebrafish. So at some point, we're interested in doing some evolutionary studies. Why is this potential lost, which we think is more cell intrinsic, possibly in the humans or mice, actually. So yeah, that's definitely an interesting comparison that we don't have an answer of why yet.
Thank you so much. Is ROS signaling necessary to drive reprogramming in neurons?
Yeah, that's a fantastic question. We do have, and we're working on a paper from Alex's lab, my postdoctoral lab, that ROS seems to be the alarm signal. What I mean by alarm signal is that the dying cells release more ROS, which stimulates stem cells. And if that step is failed, we do not get reprogramming. So it is crucial. And this is pretty well studied in many other injury paradigms, that initial cell death is, and whatever that secretes, basically, and ROS is kind of the initial one of those key damage associated molecules that people call them, is required for stimulating the wound healing or injury response in many cases. But yes, and we haven't studied this extensively in the adult, and whether if that signaling is somewhat impaired, that may affect. But given that our cells in the adult still know that there is injury, I assume it's not the step that is inhibited.
Thank you so much. I'm moving on to the next question. Brain has a highly energy needed system by its own. So if there is a dysfunction on mitochondrial homeostasis, can efficiently affect on axonal energy homeostasis. Could we clearly characterize the association between mitochondrial status and aging?
Yeah, so there's a lot of studies going on that, and mitochondrial function, or let's say motor dysfunction, is one of the key signs of aging that people use in the context of, what are the molecular differences that occurs in cells with aging. So that's absolutely true. And one thing, and again, this is kind of leading into some of the differences between our developing NEPs and adult NEPs. So in the case of the developing cells, these cells are still very immature and they haven't fully made neurons, and they use a different type of metabolism, we think, compared to the adult cells. And being more, metabolically, I don't have evidence for this, but I think it's more glycolytic metabolism, and stem cells tend to be more glycolytic and then turn on oxidative phosphorylation or mitochondrial mechanisms as they differentiate. This is kind of widely more or less studied in different stem cell contexts, which may be also why they are more permissive to reprogramming. So there's multiple layers within the stem cell differentiation paradigm. Yes, mitochondrial status is probably very critical for regenerative capacity, and also in the organ level, as the person has suggested, in terms of the aging aspect of like axonal biology and neural biology. There is lots of studies that shows that mitochondrial health is crucial for proper function.
Thank you so much for your informative answers. Do you think aging can be associated with increased inflammatory cytokines, including lymphotoxins, in the dorsal root ganglia?
Yeah, so I mean, I don't know much about directly the dorsal root ganglia, but I think generally, yes, more inflammation you have, you do worse. And the cells don't like that. And that's kind of, I didn't talk to you guys about why we're looking into microglia as well, and whether in the neonatal, in our hypothesis is during neonates, microglia, because they're actually functioning in their developmental state, is not inflammatory, where in the adult injury, actually, microglia is becoming more inflammatory, and therefore it is inhibiting regeneration. So yes, more inflammation, and this is again signs of general environmental stress or chronic stress or toxins and so on that may lead to more inflammatory like state, is never good for any types of system. Mm-hmm.
Thank you so much. I'm continuing with the next question. How far-reaching are the effects of aging and pro-youth factors in the brain?
I mean, yeah, I don't know. It's a hard question to answer. I think, I mean, the thing is depends on what we mean by pro-youth factors, right? So, whether what we're trying to stimulate. So we're trying to, for example, do a very controlled approach here, where we're hopefully trying to selectively stimulate just the stem cells or injury responsive cells. For example, there are some old studies when you force neurons to proliferate. Let's say we can use that as a possible pro-youth factor, maybe. Not really exactly what I think the person means by pro-youth factors, but most neurons die. And when we activate MYC in all the brain, for example, we only see through proliferation in the Bergman glial NEPs, which may mean that other cells, either they have other regulatory mechanisms that they couldn't induce proliferation, or they just are unhappy proliferating. So there's a lot of cell type dependent context, depending on which pro-youth factor we're talking and which cell we're talking on. So it is a little bit hard to answer this without more context, I think.
Thank you so much. Thanks for presentation. How much of the interactions between extrinsic and intrinsic regulators of neural stem cell aging could be uncovered? What are the benefits of this knowledge?
Yeah, so let's start from the second question, I guess. Benefits of this knowledge is that gives us power of regulating, right, when something goes wrong. For example, in our case, when neuron production is inhibited, if we know why is it inhibited and what factors are there, we can modulate this to induce stem cell function. To what extent this can be uncovered is, I think, this is generally, I think, biological sciences with new tools and with new developments. The extent always we uncover something that we didn't know that even was possible. And that's why, for example, in our research, we're really excited to use single cell technologies, which gave us great power on, individual cell-based, individual cell type, cell state, understanding of over time, how are these cells are functioning. So I think we don't know what we don't know, let me put it that way. But our goal in the long term is to get a full understanding of these mechanisms, but we're also limited by the technologies we can use, right?
Thank you so much again for your informative answers. Can systemic therapy approaches to brain aging reveal an age-related duality in blood based on both pro-young and aging accelerating circularity factors?
Yeah, so that's a great question. I think there is possibility, but again, our results show that it's not just cell extrinsic factors or blood. We activate sonic hedgehog signaling, for example, systemically, and we still don't get the full reprogramming. So therefore, we are more rooting for a combinatorial approach of activating different levels of signaling. And this is assuming we can overcome the cell intrinsic inhibition. So it is to be determined, but from our results, we think that it's not going to be a single magic molecule or blood transfusion that can solve this problem. Yeah.
Thank you so much again. One of the YouTube question again. Why do you think Bergman glia differentiate into GCP during injury? With less Bergman glial cells, there are any chances in tissue environments?
Yeah, so that's a fantastic question. Something we're interested in looking into. Why do I think? I don't know. It was pretty surprising for us that we decided to study this, obviously. So Bergman glia cells are a little bit special, and I didn't have time to get into their differences. They are resembling more the embryonic neuronal progenitors that are called radial glia, for example, and they start their life as radial glia and then become Bergman glia eventually. So maybe we think they have some memory of this embryonic, more what's it called, the more plastic state. We absolutely think there's role of tissue microenvironment in the pro-regenerative and anti-regenerative states. It's not really related to number of Bergman glia. I think it's more about overall the signaling and the extracellular, maybe the stiffness of the tissue or other things. And then we think why Bergman glia is the cells that are being able to regenerate is because they actually are in physical contact with GCPs during injury. So I think that's why they know what to make. But we're now testing if we stimulate, for example, ASCL1 and Bergman glia without injury, would we still get GCPs, or do we need cell death for Bergman glia to know what to make? Because we don't always get this response in other cell types. Mm-hmm.
Thank you so much. How well understood is the interlineage and cell-to-cell interactions required in vivo for expansion of different progenitors and survival of Purkinje cells?
Oh yeah, that's a great question. I didn't talk about Purkinje cells a lot today. Not at all, actually. Purkinje cells basically are kind of a central hub for all these processes we talked about. And Purkinje cells are the cells that secrete sonic hedgehog, which is required for regeneration and also just development of cerebellum. So it is, we have some understanding of some of these molecules, but we are still obviously expanding this knowledge. There's definitely interlineage communication, just as similar to the previous question I said, between GCPs and Bergman glia, and I think it's because cell-to-cell contact took part. And without Purkinje cells, none of this would have happened. But also there is the 3D structure of the cell with the layers that may be important for the migratory processes and so on. So it is, some of it we understand, some of it we hypothesize, but don't really have experimental proof. But it is absolutely crucial. And there is a lot of interlineage communication that gets into it. And one thing to note, I guess, on last bits, the interlineage aspect, the cell-to-cell, the regeneration stops when the circuitry is more established. So the cell-to-cell interaction and neurons firing may also affect the whole regenerative processes.
Thank you so much. I would like to thank you for your informative presentation and answers. Lastly, I would like to ask that what advice would you give to students studying in scientific field?
Yes, thank you for the invitation too. I really enjoyed them. Fantastic questions, everyone. I think the biggest advice is to read a lot, be proactive, talk to, try to kind of absorb as many different fields and information as you can at these early stages so that you can, decide on what excites you the most in terms of biological questions or techniques or whatever. And I think in terms of network well, and I think all the bioinformatics people I met sounds like this is part of the point of the society. So networking and getting to know people is something we sometimes don't do well, but it is important for future careers. And I mean, it goes without saying, work hard, read a lot.
On behalf of myself and all our participants, I would like to thank you for spending your valuable time, transferring your knowledge to us, and for this very informative presentation.
Thank you very much, and enjoy the rest of the talks.
Thank you so much. I wish you healthy days.
Bye-bye.